And I'll now invite your colleague. So we now have Dr. Giada Mattiuzo, also from the MHRA, and she's going to talk about standardization of COVID-19. Standardization of COVID-19 serological assays, tools, and challenges. So I'll hand over to you. Thanks, Giada.
Thank you. Can you hear me?
Yes.
Okay. Let's share my screen. And sorry, it's a little bit slow. Can you see in the presentation mode?
Yeah, that's great. Thanks, Giada.
Okay. I'm so sorry. I joined the meeting 5 minutes late. I had some IT issue, and so I don't know how much Neela has introduced us, but so I'm leading the vaccine group in the vaccine teams in the R&D at the MHRA, and the focus of my group is the development and standardization of assay used to evaluate the potency and immunogenicity of vaccine. We are based in the South Main site that some of the people I've seen in the chat and I know quite well knows as NBSC. Despite the legal change of name, we are still doing the same activity we were doing before. We are still the UK National Control Laboratory for Medicine and Biological Products, and we're still the WHO, one of the WHO International Standard, WHO Collaborative Center for Biological Standardization, and we do produce 90% of the international standards. And the talk, as I mentioned before, is focused on these standards, and these are particularly important for biological products because these are highly complex products, and it's not as important to measure how much in nanograms or milligrams you put in a tube. It's important to measure the biological effect. And because they usually produce in multi-step processes, and despite the very tight quality system in place to assure that the manufacturing is consistent, there is an inherent variability batch by batch. So it's important that we have some standards that help us make sure that the assay we're using is measuring the right thing. I will talk about standardized method a little bit later, and but the importance of having standardization is to ensure that regardless which laboratory perform the testing, we try to have very similar results, so we can come together to the same conclusion. And one of the way this is achieved, is not the only way, is by expressing the results relative to a common reference material. The WHO International Standard offers is used as the primary caliber. So what it offers is a common language that can be used to express the results to measure the biological activity. And the unit that is used is the international unit. So in this little cartoon here, there is an example of why having the same unit is important. If you want to, for example, determine the protective titer of a vaccine, and you need to compare several studies, but that comparison can only happen if everyone is expressing the results in the same units. And these slides, I'm not expecting anyone to read all of it. It's just taken from one of the British Technical Report series. It's actually explain the different hierarchy of standards. So for tertiary standards on the right, so is generally the standard you have in-house. If you buy an ELISA kit, that could be the calibrant that is in your ELISA kit. And that's the one you should use every day and you should use to report your results relative to. Then you have the secondary standards that could be general, regional, national standards. And these are very important for us because we are not expecting everyone to use international standard every day. You should just use that once to calibrate your other secondary standard or your tertiary standard. And then once the unit is assigned, then you should use the secondary standard rather than international standard. And the international standard is what makes it special is the process in which it's developed, which is very well described by the WHO in their technical report series 932. The process usually lasts between 2 and 3 years. The bottleneck is always the donation of bulk material. So my talk is specifically looking at the serological standard, and to measure antibody response, we usually prefer to have convalescent plasma serum from the individual. This is because it's very similar to the clinical sample that is tested. And sourcing this kind of material, especially in the context of an emerging pathogen, is quite challenging. The material is received by us. We do some characterization, formulation to have that stable, but that's critical step is the collaborative study. And I've seen some names, familiar names in the chat, in the list of participants. So some of you have participated to one of our collaborative study. And this is when we send the material to recommended 15 to 20 laboratories distributed globally, and ask the participant to test the standard in their assay. What we want to see is first that the standard can perform very well and can produce a dose response curve in a wide variety of assays, or at least the one that most used. And the second is expressing the results of other sample that we include in the panel that we send out. Expressing those results relative to international standard reduce the interlaboratory variation. And I'm just going to give an example. Since this is a coronavirus network, I'm going to use the example that for the development of the first international standard for MERS antibody. This project started in July 2019, and it was a collaboration between ourselves and the International Vaccine Institute in South Korea and CEPI. IVI was working with the Seoul National University to identify MERS recovered patients, and then they screened quite a few of them, and they managed to find 4 of them that still have high level of antibody against MERS. Through several collections, we received 250 ml of serum from each of these individuals. We usually perform a solvent detergent treatment. This is a procedure that has been used for 30 plus years to enhance the safety of blood-derived product. And then we assess the neutralization antibody titers that were correlated very well with the antibody titers detected by IVI. And then we pull the sample. We created 2 standards. One was the candidate international standard, which is actually in our catalog now as 19178. And then we had a working reagent. This can be used if you want as a secondary standard, and is again in our catalog. And we also, as a part of the collaborative study that we launched, included the 4 individual samples. So as I mentioned, this collaborative study started in January 2020, and I have to say it has been incredibly challenging because of the COVID pandemic that happened at the same time. Nevertheless, we managed to find 11 participants that had the time and willingness to help us out and test the standard in their laboratory. Overall, the standard was tested in 10 ELISA, 3 neutralization assays using the authentic virus in BSL-3, 6 pseudo-neutralization assay with a lentiviral vector expressing luciferase, and then 2 immunofluorescent assay and 1 immunofocus assay. And this is an example of the results. I'm just focusing on the neutralization results, but similar pattern can be seen for the binding. And on the left side, you have the results from each of the sample, which are on the x-axis, as reported by the participant in terms of 50% neutralization titer. And there is quite a wide distribution of the results between laboratories for each of the sample. When you use the CAD International Standard, which was the sample S55, you have the results on the right, in which most of the laboratory arrived to a very similar value expressed in international unit, even when different strains of either spike or virus have been used. The table underneath the graphs are another more statistical way to report the reduction of interlaboratory variation by looking at the coefficient of variation, which is greatly reduced when you express the results in international unit. As mentioned, the standard was developed in 2020 and established by the expert committee in biological standardization of the WHO in their meeting in October 2020, and is available in our catalog. So that's how we made the standard in more or less peace condition. So you select your partner, you develop the standard, collaborative study. But what happened when we needed to speed up these timelines? And the example that happened to everyone, I guess, is the pandemic, COVID-19 pandemic. So between January and March 2020, when it was declared by the WHO public health emergency of international concern, we started to develop assay and reagents for responding to the pandemic and supporting mainly vaccine developer and for development of therapeutics. And this is a couple of publications with several collaborators in UK, which we have contributed to, mainly looking at the production of pseudotype neutralization assay. And in end of March 2020, we made available a research reagent for SARS-CoV-2 RNA. This is to support the diagnosis of the molecular diagnosis of the virus infection. And then in April, in collaboration with NHSPT and supported by SEPI, we made a reference reagent for anti-SARS-CoV-2 antibody. And that was high titer convalescent plasma from one UK recovered patient. So the reason why we made this research reagent is because we knew that the production of international standard will take longer time. And there was a need to have a common reagent then and there to actually not only help compare results between lab, but also to have some laboratories setting up their own assay. And that's why we also made a reference panel with the five panel member. One was a negative and 4 of them were different titer of antibody against the SARS-CoV-2. The research reagents was then included in the collaborative study for the establishment of the international standard, and it was calibrated to the standard. The reason we did that is because everyone that was using this research reagent to report the results relative to it, once the international standard has been established, they could quite quickly translate their data in international unit by using that value. As I mentioned, the two collaborative studies started in July, one for the antibody standard, one for the RNA standard. And those were both established by the WHO-CBS in an extraordinary meeting in December 2020. So we had the first WHO international standard for SARS-CoV-2 RNA, one for the immunoglobulin, and a reference panel, which was very similar in terms of antibody activity to the 2018. And together with colleagues at SEPI and at the WHO, we did promote the use of the standard, trying to explain why the using the standard is important. And we were so successful that 8 months later, we completely deplete our stock of the antibody standard. And this highlighted a challenge, which is how do we keep the international standard relevant, especially when, like SARS-CoV-2, we have an ever-changing scenario and the introduction of the variant of concern. So the next standard we developed, instead of one standard, we made two. So the second international standard is a like-for-like replacement of the first international standard. The material used is very similar, which is convalescent plasma for individuals infected early 2020. And the reason we did that is because we didn't want that the people that have been using the first international standard all in a sudden have to redo all their calibration because we have a new standard there. So we need to maintain the continuity between international standards. Issue with this standard, which is the same for the first, was that it has very low or no potency in terms of neutralization activity against the Omicron variants. And that's why we made the second standard, which is the first WHO international standard for SARS-CoV-2 antibody for variant of concern. And this is a convalescent plasma of individuals that have been infected with some of the variant and then vaccinated. And looking at the literature and by our own experience, we have seen that this gives you a broader response than just infected individual. And indeed, despite that this material does not contain any convalescent plasma from Omicron-infected individual, in the collaborative study, we have seen a very good response to the variants that were available at the time, which BA1 and BA2. And the intended use of this standard is to be used as a calibrant for neutralization assay for variant of concern. And how good it is with new variants. So I apologize, some of this data may be a little bit dated. So we did in-house pseudo-neutralization assay and the data from Catherine and Emma, and we received the spike protein from the G2P UK National Virology Consortium for Q1.1 XBB. And then Jan Ledaf, Giada MHRA, as part of the CEPI-funded Agility project in collaboration with UK-ICN, has tested the standard against XBB 1.1 and 1.5. And I think the data I have here is probably done only once, but they may have more recent data. And then we ask Ruth Harvey and Mary Hu at the Crick Institute to do again neutralization assay with real virus for XBB, and that's the results. We also distributed this standard as part of an international network organized by the WHO SARS-CoV-2 Immune Escape Working Group as the common reagent to compare the results between the network. And very briefly, I just want to highlight some of the misuse of the standard. So in the past, I've seen people thinking if you use the international standard that you standardize your method, That's not the case. I'm just going to show later what standardization is. It's not validation tool. You need a panel for that. And we strongly discourage everyone to use that as a positive control because we will not be able to supply that large amount. The other important thing is it will not make a bad assay good, so and it will not help harmonization of two assays that do not have a good correlation. So if two assays rank sample in a different way, the use of the international standard will not make the two assays all in standard ranking the sample in the same manner. The way in which the standard works is if you have two assays, and that's in the graphs here, and one has consistently higher results than the other assay. By reporting the results in international unit, you just move yourself towards that perfect correlation line. And the standardized method I mentioned a few times. So to actually fully standardize assay, the use of common reference reagent helps and is important, but you really need to share SOP and share critical reagent. And an example of this is given by the CEPI Centralized Laboratory Network, which was developed in response to COVID-19 and is now moving to the next phase for other epidemic and pandemic threats. And as I was also as part of the network. And very briefly, since we're covering all the coronaviruses, it's very difficult to find the material for each of the 4 seasonal coronaviruses because of their high prevalence. But to complete the picture of the outbreak ones, we are currently conducting, about to launch a collaborative study for the SARS-CoV-1. So we've been lucky enough to be provided by the NIA the 500 ml of purified immunoglobulin G which were manufactured under GMP for therapeutic use in response to the SARS-CoV outbreak. These were prepared from convalescent plasma collected from individuals from Hong Kong. And the state of the project is we have the material in-house. We are about next month to formulate in a stabilized manner and to launch the collaborative study at the December or January next year. And the study organizers, if anyone is interested and would like to participate, is Emma Bentley, and her email address is here. And finally, for the acknowledgements, well, there is a lot of people at MHRA that have contributed to all these projects. Our biggest support is our relationship with CEPI that has been going on since 2018 and has been amazing. And then all the groups that in a way or another have donated material, and obviously all the anonymous donors and the collaborative study participants. And that's me. Thank you very much.